Hi, welcome to Biology classes with Lakshmi Chandra. In today's session, we will discuss on the first step in respiration, that is glycolysis. So this is the first and the primary step in cellular respiration. Do you know who found out the steps in glycolysis? Three scientists, Gustav Emden, Otto Meyerhof and J. Parnas. They were the scientists who were the pioneers in finding out the various steps involved in glycolysis. So, this glycolysis is also called as EMP pathway. That is letters taken from the first letters of their names. EMP, Emden, Meyerhof, Parnas pathway. This glycolysis, as the name suggests, is coming from two Greek words, glykos and lysis. Glykos relates to glucose and lysis means splitting up. That is splitting up of glucose. For what? Why this glucose is getting splitted up during cellular respiration? As you know, respiration is basically required as a process which releases energy that is stored in the food materials that we all consume. Now we will be consuming carbohydrates that is getting digested into small glucose fructose units, and these are absorbed by our blood, and they are carried to various cells of our body. Now what happens? This glucose enters into a cell. Inside the cell, what happens? This glucose has to break its bonds and release the energy so that the cell gets that energy and it starts doing its functions. So that basic step we are going to learn in glycolysis. So the glucose has to enter into the cell first. But after entering into the cell, in no way the glucose should come out. So what happens? The glucose gets converted to glucose 6-phosphate. Glucose 6-phosphate. What does this mean? Glucose is having 6 phosphates with it? No. It means that glucose, glucose, when you write glucose, you write like this, C6H12O6. That means glucose is a molecule having 6 carbon atoms. Out of these 6 carbon atoms, in the 6th carbon atom, 1 phosphate is getting added. So here, this 6 refers to the position of the carbon atom in which the phosphate is getting added. To this glucose molecule, in its 6th carbon, phosphate is getting added. That is glucose 6-phosphate. Why this step is happening is because when you have a cell and inside the cell, a glucose molecule may enter through some channels. Okay? So as it enters into the cell, it should not come out. It has to be trapped inside for the release of energy through cellular respiration. So what happens here is this glucose gets attached to a phosphate molecule in its 6th position. It becomes glucose 6-phosphate. Phosphate, when it is getting added, it becomes a polar molecule, and polar molecules cannot easily come out of the cell. So this forms the first step of splitting of glucose, that is glucose is getting converted to glucose 6-phosphate. And you can see here, this phosphate molecule has to come from somewhere, isn't it? So from where that is coming? It is coming from the ATP. I hope you all know about this molecule, ATP, adenosine triphosphate. It is called as the energy currency of this cell. If the cell has to do some activities, it has to spend this ATP. That means it has to utilize the ATP. Here, what for this ATP is used? adenosine triphosphate. 3 phosphates are there. Out of that, 1 phosphate is taken and added to the 6th position of this glucose molecule. So what happens to the glucose? Glucose will become glucose 6-phosphate. And what happens to ATP? ATP is losing 1 phosphate. So instead of triphosphate, it is having only 2 phosphates. So how do we say? It becomes ADP, adenosine diphosphate. And this is not an easy reaction. For this, there is help of an enzyme called as hexokinase. Hexokinase. Let me just tell you here, see, hexo means 6 carbon compound. To the 6 carbon compound, what is getting added? A phosphate is getting added. So whenever you see the enzyme name kinase here, what this kinase does is, it will take phosphate, it will transfer phosphate from a high energy molecule to another molecule. So here phosphate is getting transferred from a high energy molecule, that is from ATP, it is getting transferred to glucose. So this addition of phosphate is done by the enzyme kinase. Since here it is adding phosphate to a 6 carbon compound, this enzyme is called as hexokinase, and there are different categories of hexokinase. To be specific here, we can say it is glucokinase. So difference between this hexokinase, glucokinase and their activities will be dealt with in another session. Please wait for that. So I hope you all have familiarized with the first step in glycolysis, that is glucose is getting converted to glucose 6-phosphate. Now let us move to the second step. The second step is a reversible step. Glucose 6-phosphate is getting converted to fructose 6-phosphate. So here you can see the only change is glucose getting converted to fructose, and both of them are isomers. So the enzyme involved here is isomerase. Now comes the next step, a very important step. Fructose 6-phosphate is getting converted to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate, or it is also in some books it is given fructose 1,6-diphosphate. Whatever it is, I hope from this you can understand. Fructose 6-phosphate means the 6th position of fructose, phosphate is getting added. So can you guess what does fructose 1,6-bisphosphate means? Exactly. It means phosphate is added to the 1st position of fructose also. So how many phosphate molecules are there? 2 are there, isn't it? That is why here it is mentioned it as bisphosphate. Instead of this, it can also be written as diphosphate, Di. That means 2 phosphate molecules are there. Where are they? One in the 1st position of fructose, 1st carbon, and the other one in the 6th carbon it is attached. So one more phosphate is attached, isn't it? So where it will get another phosphate? Again, like here it has got from ATP. In this step also the phosphate is coming from ATP. One phosphate is coming from ATP. So what happens to ATP? Instead of triphosphate, it will become a diphosphate. So here also what is the reaction? Phosphate is taken from a high energy molecule and added to another molecule, isn't it? So here again, what will be the name of the enzyme? The name of the enzyme is going to be kinase only. But it is not hexokinase, isn't it? Then what is it? You can say it as phosphofructokinase. So it is very easy to know the names of the enzyme. From the substrate itself you can say phosphofructokinase. Phosphofructokinase. That is the name of the enzyme. Let me write here properly. So whenever there is addition of phosphate from a high energy molecule to another molecule, please remember it is kinase enzyme. you can just relate it to the name of the substrate. So it is very easy to name the enzyme. Here it is phosphofructokinase. You can just write this as PFK. So it is specifically saying it is phosphofructokinase 1. You can write it as PFK1. So the enzyme names, let me just mark it with a blue marker. Hexokinase here, isomerase here, phosphofructokinase 1. That is converting fructose 6-phosphate to fructose 1,6-bisphosphate. Now this step is very important step. This is called as the rate limiting step. Okay, so my dear students, you should know about this, the meaning of a rate limiting step in any kind of reaction. This step is also a committed step in glycolysis. So why this is called as the rate limiting step? Rate limiting step because this step is the slowest step in glycolysis. So this actually tells the rate in which the reaction will move forward. Okay, and it is also called as the committed step. These steps that we have seen here, we can see that in these two areas ATP is getting utilized or energy is getting utilized here. Actually, cellular respiration of this glycolysis, it is taking place to release energy, but here we can see that energy is getting utilized. So this first phase of this glycolysis where energy is getting utilized, this phase is called as energy investment phase. Energy investment phase because two molecules of ATP are getting utilized. Now we have reached till here, till fructose 1,6-bisphosphate. Now what happens to this fructose 1,6-bisphosphate is, it gets splitted up into two molecules, two different molecules. Okay, so far we know glucose is a 6-carbon compound. Glucose 6-phosphate is also having 6 carbons. Fructose 6-phosphate, fructose 1,6-phosphate, all these molecules are 6-carbon compounds. Now here it gets splitted up into two molecules. So each of these two molecules will have how many carbon atoms? 3 plus 3 each. Okay, so here what happens is a 6-carbon compound is now getting splitted up into two molecules of 3 carbon each. Let us know the names of those molecules. They are called as dihydroxy acetone phosphate, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now don't just get frightened by looking at the name. Let us come to some easy step. This you can just call it as DAP, DHAP. Okay, and this we can call it as GAP, GAP. Glyceraldehyde 3-phosphate. 3-phosphate means what? Does it have 3 phosphate molecules? No. Means in the third carbon phosphate molecule is attached. Now here dihydroxyacetone phosphate. Here there is no number mentioned. Means what? Where is the phosphate attached? It is attached to the first carbon. So first and third. Here it is attached to the first carbon. Okay. Here phosphate is attached to the third carbon. And both these molecules are having how many carbon atoms? Both of them are 3 carbon compounds. They are 3 carbon compounds. What kind of compound was glucose? Glucose had 6 carbon atoms. Now it got splitted up into 2 molecules, each having 3 carbon atoms. So glucose is having 6 carbon. You call glucose as a hexose. Hexose molecule, a sugar molecule having 6 carbon atoms. Now what will you call these two sugar molecules having 3 carbon atoms? You call them as a triose. So we can say hexose is getting splitted up into 2 triose here. So this glycolysis is also called as triosis. Triosis. Earlier I told you another name. It is also known as the EMP pathway. Embden-Meyerhof-Paranas pathway. Now I am telling you it is also called as triosis because the 6 carbon glucose is getting splitted up into 2 3 carbon compounds, and of course there is involvement of an enzyme here, and that enzyme is called as aldolase. Aldolase. This is the enzyme which is splitting up fructose 1,6-bisphosphate to these 2 compounds, DAP and GAP. Now interestingly, these 2 molecules are also interconvertible. They are also interconvertible. And they are isomers also. They are 3 carbon molecules, they are interconvertible, they are isomers. So which enzyme is doing this interconversion? It is done by triose isomerase. Isn't it very easy to remember all these things? 3 carbon isomers. So the enzyme involved is triose isomerase. They are converting it. And you know, even though these 2 are interconvertible, dihydroxyacetone phosphate is getting converted to glyceraldehyde 3-phosphate, and every time the further steps proceeds through this region only. That is, what happens to glyceraldehyde 3-phosphate next? So always this aldehyde here is getting converted to subsequent molecules. And so always whenever DAP is produced here, it is converted to glyceraldehyde 3-phosphate, and then the steps proceeds. So here, dear students, what you have to understand is one glucose molecule got splitted up into 2 molecules. After that, whatever steps I am saying to this glyceraldehyde 3-phosphate, whatever happens to glyceraldehyde 3-phosphate, the same step actually is repeating twice here. Why? One is with the actual glyceraldehyde 3-phosphate. Another one is this DAP is getting converted to glyceraldehyde 3-phosphate, and the same steps continues for glyceraldehyde, that converted glyceraldehyde 3-phosphate. So whatever I am going to say next actually happens for 2 molecules. One is DAP, another one is GAP. Here, glyceraldehyde 3-phosphate is now getting converted to 1,3-bisphospho glyceric acid. 1,3-bisphosphoglyceric acid. So my dear students, just note the change in these 2 compounds here. Here you can see that this is an aldehyde which is getting converted to an acid molecule. And another change here, it is phosphate, there is only 1 phosphate which is added into the 3rd carbon. Here you have 1 more phosphate, that is bisphosphate. That means 2 phosphates are there, one already in the 3rd, another one in the 1st carbon. So what should be the changes happening here? Aldehyde should be converted to acid, 1 more phosphate have to be added. So I know, whenever you see here in these 2 steps, when phosphates are getting added, I told you kinase is the enzyme and ATP is actually doing that. But in this step, there is a slight change. Here, the phosphate is not coming from ATP and there is no involvement of the enzyme kinase. Instead, here the phosphate is coming from an inorganic phosphate, like phosphoric acid. It is coming from an inorganic phosphate and it's getting added into the 1st position. That's how 2 phosphates are coming. Now the next change I told you is aldehyde is getting converted to acid. Here, dear students, please understand, whenever an aldehyde is getting converted to acid, a particular reaction should take place. That is oxidation. Aldehyde is getting converted to acid through an oxidation reaction. And what is this oxidation? Addition of oxygen. But you know, glycolysis is actually a universal step in respiration. This part, dear students, you should understand very well. Glycolysis is a universal step in respiration. That means it takes place in all kinds of cells, whether it is a prokaryotic cell or a eukaryotic cell, an aerobic cell, an anaerobic cell, doesn't actually mean anything to this glycolysis. It takes place in all kind of cells. So, as I mentioned, it can take place in aerobic as well as in anaerobic cells. In anaerobic cell, it won't take oxygen. So, oxidation, as I've told you, it is addition of oxygen. So, in this step, conversion of aldehyde to acid cannot take place by the addition of oxygen. Then how it will take place? Oxidation is also removal of hydrogen. So, what should happen here? Dehydrogenation. Hydrogen have to be removed. So, naturally the enzyme that has to come here is which one? Dehydrogenase. This enzyme will remove hydrogen and will convert glyceraldehyde to an acid, 1,3-bisphosphoglyceric acid. And the hydrogen that is getting removed is accepted by a universal hydrogen acceptor. Which is that molecule? The universal hydrogen acceptor? It is NAD. So here what happens? NAD, what is this NAD? It is a universal hydrogen acceptor. Nicotinamide, adenine dinucleotide. NAD, nicotinamide adenine dinucleotide. It is a universal hydrogen acceptor. So the hydrogen which is removed, dehydrogenase enzyme is coming and it is removing hydrogen from the aldehyde group. Okay, and that hydrogen is accepted by the NAD and NAD gets converted to NADH. It gets converted to NADH, and this NADH has got a significant role if the cell is doing aerobic respiration. That portion I will deal with it in the coming videos. Definitely I'll deal with that. And that is very important as well. When a cell is doing aerobic respiration, what happens? Okay. So in glycolysis, we'll move further. So here what happens is glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglyceric acid by addition of an inorganic phosphate, removal of hydrogen likewise. Now next what happens is this 1,3-bisphosphoglyceric acid is getting converted to 3-phosphoglyceric acid. You can, instead of glyceric acid, you can also use the word glycerate. 1,3-bisphosphoglycerate, 3-phosphoglycerate, like that also you can use. Okay. So here see 1,3-bisphosphoglycerate converted to 3-phosphoglyceric acid. So what happened here? One phosphate is not found. Where is that phosphate gone? Yes. Now comes the actual area where energy is getting produced. Energy is getting produced means what is that energy actually? In the form of ATP. So the first ATP production takes place here. So this phosphate is taken by ADP and this ADP is getting converted to ATP. ADP becomes ATP. From where that ADP is getting a phosphate? From the substrate it is getting the phosphate and the ATP is getting produced. So this area is producing an an ATP molecule with the help of a substrate. So this step you call here ATP is produced as a result of substrate level phosphorylation. Phosphorylation means phosphate is getting added. From where it is coming? From the substrate. To where it is added? To ADP it is added. So what it becomes? It becomes the energy molecule, that is ATP. Now dear students, please remember here one thing. Dihydroxyacetone phosphate is also getting converted to glyceraldehyde 3-phosphate, and these steps are proceeding. So actually there is how many glyceraldehyde 3-phosphate molecules are here? One the actual one, one the converted one. So actually there are 2 glyceraldehyde 3-phosphate molecules. So there are 2 molecules of 1,3-bisphosphoglyceric acid formed. So there are 2 molecules of ATP that is getting formed here. So you see here in the first 2 steps ATP was getting utilized. 2 molecules were getting utilized. Here 2 molecules are getting produced. So what is the net gain now? 0, 0. Isn't it? Minus 2 here, plus 2 here, 0. So far actually energy the cell did not get the energy. Let us see what happens next. So 1,3-bisphosphoglyceric acid is converted to 3-phosphoglyceric acid. Now the story goes on. What happens next? This is getting converted to 2-phosphoglyceric acid. Again you see these 2. There is only change in the position. So this is just the matter of a positional isomerization. 3-phosphoglyceric acid is getting converted to 2-phosphoglyceric acid. So it is just a matter of positional isomerization. So you can just use the enzyme here is mutase, a mutation reaction, just a positional isomerization. Now dear students, what about here this step, which is the enzyme that is coming here? Is there an addition of phosphate molecule? Yes, there is addition. Phosphate molecule is added to ADP, isn't it? And it comes to ATP. So when there is addition of phosphate from a high energy molecule, what happens? Which is the enzyme there? The enzyme is kinase. So which kinase is this one? For which kinase? That you can just get from the name of the substrate, isn't it? Phosphoglycerate kinase. The name of the enzyme is phosphoglycerate kinase. So there is nothing very difficult to know the name of the enzymes. Just from the substrate itself you will come to know about the enzymes. So glycolysis is nothing, there is nothing to by heart here. Just understand the steps and move forward with a logic. Okay. So 3-phosphoglyceric acid is converted to 2-phosphoglyceric acid. Now this 2-phosphoglyceric acid is getting converted to another molecule called as phosphoenol pyruvate. Phosphoenol pyruvate. Phosphate is there. One more thing is coming, enol pyruvate. Okay. So here what happens is, here there is the enzyme involved is enolase. Enolase is the enzyme involved here. And one more thing happens, water molecule is getting eliminated. So what is this step? Dehydration. Water molecule is getting eliminated, dehydration. Here what was the step, children? Dehydrogenation. Hydrogen is getting removed. Here what is getting removed? Water, dehydration. Okay. And all these things, it is actually done very fast with the help of the enzyme enolase. So mutase the enzyme here, enolase the enzyme here. Now this phosphoenol pyruvate is now getting converted to pyruvate. Pyruvate, or you can say pyruvic acid also. So see here, there is no phosphate. Phosphoenol pyruvate to pyruvate. So one phosphate is not here in the final product. So again, where is that phosphate gone? Where is that phosphate gone? That phosphate is again taken up by our ADP. That phosphate is taken up by ADP. So ADP takes up that phosphate and it becomes the energy currency ATP, isn't it? So again, which will be the enzyme involved here? Kinase enzyme. So the name of the enzyme is kinase. So you can write here pyruvate kinase. Pyruvate kinase is the enzyme. Now here again you can see here, children, phosphate is coming from the substrate, and as such ATP is getting generated. So this is also a step where you can see substrate level phosphorylation. Substrate level phosphorylation. So here 2 ATP molecules are produced as a result of substrate level phosphorylation. The same thing continues. Here also 2 ATP molecules are produced as a result of substrate level phosphorylation. How this 2 comes? One is coming as a result of the reaction of glyceraldehyde 3-phosphate is producing ATP. Another one, DHAP is converted to glyceraldehyde 3-phosphate and that is also producing ATP. Likewise, 2 ATP molecules are produced here. So what is the net gain of ATP in glycolysis? It is 2 ATP. Net gain of ATP in the case of glycolysis is 2. This is true in the case of anaerobic cells. If this is an aerobic cell, children, what happens? This particular molecule, NAD and NADH. This NADH enters into a very special chain called as electron transport chain. As a result, that NADH will also produce ATP. Every NADH will produce 2.5 to 3 ATP molecules and NAD is getting produced back. That is another session which can be dealt with later in detail. So here I hope you understood how glucose is getting converted to pyruvate. And what happens to this pyruvate? What is the fate of the pyruvate? Depends upon in which type of cell it is. As I have told you, glycolysis is a universal step in respiration. It is a common respiratory pathway in all organisms and this takes place in the cytoplasm. There is no need of any cell organelle because all these enzymes that I have mentioned here, they are present in the cytoplasm of every cell. So this process takes place in the cytoplasm. So glycolysis is also called as cytoplasmic respiration. Now what happens to pyruvate will be dealt with in the coming session. Thank you for watching. Keep asking your doubts. Keep supporting. Thank you all.